right. Well, welcome to the June 2025 StaPH-B Talk. First StaPH-B Talk in a while, but we're back. If anybody doesn't know me, my name is Abby. I work at the Utah Public Health Lab with Kelly, and it's kind of been my mission to revive StaPH-B a little bit. So I've started with some of the StaPH-B talks. So very excited. We're back up and running. And so today we have Logan Fink, who most of you know, here to talk about the creation of bioinformatics SOPs in public health laboratories. So Logan, if you want to get started.
Thing. Well, hello everybody, and thank you for having me on. This is exciting to be on the other side of the Staff B Talks. And thank you to Abby for getting this all kind of up and running again. It's really exciting to see this kind of moving forward. So yeah, I'll go ahead and jump right into it. Um, as a bit of a disclaimer before I begin, I wasn't sure if I was going to be the whole hour or if I was going to be sharing this hour with some other people. So I might not take up the full time. Um, but I figured it might be good because we might have some time for kind of discussion and stuff at the end, just because I think that there's probably a lot that people have to say on this particular issue. So um, I'm gonna go ahead and jump into it. So this talk is going to be on the creation of SBG, which is our Sequencing and Bioinformatic Group's Bioinformatics Quality and Processes SOP. Um, so let's go ahead and get started. So I figured I'd start a little bit with the background. I think, this group probably has a pretty, uh, good idea of what the limitations are of bioinformatics in public health, but um, just in case anybody needs a refresher, I think we can go over some of these different things. So I've made the statement here that bioinformatics is not a new field, but regulatory guidance has been kind of minimal in terms of bioinformatic analysis and testing. So even though bioinformatics is not really new, especially in public health, I feel like we have been a little bit slow to kind of like adopt some of the kind of aspects of laboratory method development and validation and testing and things like that that normally kind of accompany wet lab and science kind of principles within other aspects of the laboratory, but they've been a bit lacking and maybe less well defined in the field of bioinformatics. And so this has been something that I think a lot of people have been kind of thinking about for a while. And I think a lot of people are trying to look to some of these different kind of regulatory bodies and people who are typically tasked with, providing some of this kind of regulation and guidance. In order to kind of come up with, what it is that they want to adopt at their own laboratories. But the guidance has been vague and minimal in a lot of different areas. And I'm not going to get into too many details and specifics. I would say probably in terms of like clinical testing in public health, as we're starting to, kind of come into the mainstream with, bioinformatically derived results, this has been something that a lot of people have now started to kind of pay a little bit more attention to, and we have to kind of come up with ways that, we can ensure that these results are meeting the standards of, the public health regulation that we are tasked with upholding. And also kind of adapt it to, something that is not necessarily something that we've always seen um, performed, or it's a little bit of a different kind of method. And so I think with like CAP and CLIA, CAP has some pretty good guidance in terms of like the bioinformatics kind of like regulation that they recommend, but unfortunately with like CAP labs, the that is all pretty much kind of like kept within the laboratory. It's not really widely kind of distributed beyond the CAP labs. And CLIA has had they have some kind of guidance that you can find a little bit here and there, but I would say a lot of the guidance that I've seen places regulations heavily kind of on data handling and that kind of aspect of bioinformatics, and maybe not necessarily as much on kind of like, method development and on kind of the practices of actually adopting bioinformatics within a laboratory or providing, guidance as to how, these kind of principles should be kept. So I, as another kind of I guess bullet point here, I also have so there's been a couple of other groups who are kind of working towards creating some guidance around like documents and things like that. Like NGS subcommittee had some guidance around data retention policies and things like that, and then the CDC NGS quality initiative has also provided some guidance in terms of that they have kind of a trove of documents that are available to adapt and to be able to come up with, ways that you want to use kind of like the templates that they have provided in your laboratory. But I think a lot of that is left up to the individual user and the individual laboratory that then decides to adopt those. And so um, that's just kind of some of the background that I'm uh, kind of going into this with. And so for us here at DCLS, I would say that we had kind of a minimally defined guidance around the creation and maintenance of bioinformatics software. And I don't think that's, any shade to us. I think that that is something that, a lot of people didn't necessarily have, kind of well-established guidance and well-established kind of criteria around those particular aspects of bioinformatics. Some people did, and they're amazing. But I think a lot of us are kind of like in the same kind of boat in that as we're kind of moving forward in this field together in public health, we're kind of realizing, oh, we need to kind of catch up in some areas. Um, and I would say that for us, multiple aspects of development and upkeep were kind of under these, de facto best practices. So it was kind of things that we were just doing in the lab, day to day. I don't think that our bioinformaticians were necessarily doing anything wrong, but we just didn't have like a standardized way to kind of talk about how we were going to be doing maintenance, what we were going to be doing for you know, creation and upkeep of bioinformatics pipelines. And so we thought that, if we don't have something that is really well defined, this can lead to inconsistencies in application. It can lead to people not knowing, what the practices are. And especially when new scientists are brought on, we wanted a way to kind of like bring them into the fold, so to speak. And we're kind of working on some other documentation to as kind of like a, new bioinformatician packet. So when you come into the lab, you have kind of like a step list of different things that familiarize yourself with. And then also, this would be included in that as part of the requirements that, any new bioinformatician brought into the lab would be reading this SOP, so that they're, coming in with a basis and that they know, what we do here at DCLS and how we promote our bioinformatics. And this all kind of came about because of some current occurrence managements. And so, we have some kind of very quality-minded leadership. And so, we had an occurrence management that happened with a piece of software that was developed at DCLS, and then that kind of led leadership to scrutinize it a little bit more. And they were kind of saying, like, well, who's reviewing these kind of things? What are the actual, like standardized processes that are in place in order to, do this kind of development to do make sure that mistakes aren't making their way through in the pipelines and the codes and things like that. When you find a mistake, what is the process for fixing that and then creating a patch or a solution in its place. And so then that did kind of lead to a revamping of our practices. All right, and so our solution was so our bioinformatics group sequencing and bioinformatics group manager, asked the bioinformaticians to draft out specific guidance to address the standard operating procedures of the bioinformaticians in our group. Um, and so we all met in person over the course of a few months on a scheduled basis to establish the document. And so this was all kind of, led by the group. This was something that, we were all putting our heads together and trying to come up with, how do we uh, tackle this? And so we decided on the scope and the component sections of the document together. It was a process of back and forth, and there were, you know, some things initially got created and then were scrapped, and some things didn't make it to the final, cut, I guess. Um, but we did kind of ultimately decide that instead of getting super duper specific with the rules, this document should be more of a guide to the practice. And so this is something that's going to be a little bit more general as opposed to more specific. So we decided that that was probably the best way to go, especially with, kind of like an overarching document of this nature. Um, because we wanted it to be, accessible, but, not necessarily unwieldy. If you're coming into a new job and then they give you this, bioinformatics handbook that's, 500 pages and they're like, okay, read this by tomorrow. That's going to be a little bit overwhelming. So we wanted to make this at least a little bit more general, and then we can get into the specifics about some of the different kind of components later. And so this was our this is just kind of like the overview of the document itself. And I will apologize. This talk is probably not going to be one of my more like engaging or exciting talks. There's a lot of regulatory documents and kind of explanations of this particular SOP. And so for the I guess the quality minded people in the audience, maybe this is like super exciting, but I apologize, my slides are a little bit bland on this, but I think that it is important. Um, so the bioinformatics SOP is laid out into kind of five major sections. So we went through this kind of scope first. We went through, environments and versioning definitions, then verification and validation, development of standard practices and expectations, and then tool maintenance. And there's a couple of additional sections that are at the end of the document for, the references, and there's some appendices which kind of go through some of the a little bit more detail about some of the kind of like development practices, the tool maintenance and things like that. We have a couple of images and things that help to kind of define those a little bit better. And so all in all, the document was 13 pages, which I think is reasonable. Like again, we were trying to make this accessible and something that would be somewhat comprehensive, but not unwieldy. And so 13 pages, I think, is hopefully an acceptable kind of length. All right, so getting into the kind of specific sections of the document that we created here, and again, as kind of a caveat, this is what we came up with here at DCLS, but this is in no way meant to be prescriptive or meant to signal that this is the only way that you could create a document of this nature. I think that we were just trying to come up with what made sense to us here, and this is kind of what we came up with. But I would encourage anybody who is thinking about kind of creating something like this for their own lab to look at your own practices, look at your own team, and really come together to come up with these sections, because that's, I think, what ultimately is what created a good document for us, was that we had the buy-in of the bioinformaticians and the people who were on these teams, actively contributing to the creation of the document. Um, and so it outlines kind of these standard practices across bioinformatics, um, with us at DCLS, and it gives some guidance. So in this beginning section, we did kind of, try to say that this is going to be general, but it will get into more specifics with appropriate SOPs and job aids for individual tools. And so, anything that gets created at DCLS then has to have documentation for that specific tool. And so we're getting into more of the specifics into those kind of things. And so that's, this document isn't going to get into the specifics of everything. Um, but for the purposes of this document, a tool was defined as any product created by the bioinformatics team. So this includes, but is not limited to, pipelines, databases, applications. Um, we just it got a little bit confusing as we were going through the document because some people would say pipeline, some people would say database, some people would say like tool, some people would say certain things, and so we tried to kind of like streamline the language, standardize it, so that it was just we were using the term tool to denote anything that was created by the bioinformatics team, and so then that's what this section is kind of all about. All right. And so this section went into the environment and versioning definitions. And so a lot of this stuff was just kind of trying to explain like what are these things? What are we saying when we have a development environment? And for us, that is going to be anything that has passed through uh, is going to be the beginning kind of stages of a tool. And so when we have a development environment, that is kind of like open for us to play around with, to make modifications, to make mistakes, to do testing kind of minimally as we're kind of developing this, making sure that it works. And so the development environment then would lead into the testing environment. And so we want to make sure that we have environments that are specific for these kind of different phases of development. And the testing environment, we would want to mimic the production environment exactly, but that would be used for kind of like a final test, so making sure that before you put it into prod and make sure that it's going out to the world basically, you have a test environment to make sure that you can test everything, make sure that everything works. If you do find any bugs or anything, then you can bump it back down to the dev environment and then fix those kinks, fix those bugs, and then go back up to the testing environment. Once it passes through the testing environment, then it would go into production. And so the production is, considered more of like a locked down environment. And so that is meant to be more of a way that, we're not touching it once it goes into production. So, production is stable. Production is what we use to then actually run the pipelines. We also kind of define, what is version control. So probably I don't need to define that for the people on this call. But we have some kind of definitions and things like that. We also go into a little bit of, what are the different repositories that are used by bioinformaticians at DCLS. And yeah, so going over that again, A, B, and C are the types of environments, and cover a few scenarios in which they would be used. So kind of, some of the things that I talked about. And then with version control, this was a really difficult section, I will say, to kind of define and come up with, and we went through a bunch of different kind of iterations, and I'll get into it a little bit more, but with version control, we decided to kind of go based on what we thought would be a major change, a minor change, a patch, and then the semantic versioning is kind of that picture that you see in the bottom right corner here. And so for us, I actually have it in the notes here just so that I can head off any questions about like what's a major change, what's a minor change. So for us, the way that we defined a major change, and it's pretty high level, I would say, is a change to a tool that adds new information or features which directly affect the generation of results or breaks backwards compatibility in a significant way. And of course, backwards compatibility is defined as being compatible with previous versions of the tool. And we get into a little bit more about the major changes and what those kind of entail in another section. So I'll kind of leave that off for now. With a minor change, we said that's a change to a tool that does not directly affect the generation of results, and has minimum impact, if any, on backwards compatibility. For details on minor change verifications, we also have more information in one of the later sections. Um, and so you know, it it does get to be a little bit tricky sometimes, what counts as a major change, what counts as a minor change, uh, and then uh, how do you, version those as you go forward. With the semantic versioning, we have this handy diagram that I think is really great for use in terms of kind of, any sort of like version control as we go forward. And then, of course, the repositories in use at DCLS. So typically for us, we're contributing to Docker Hub and GitHub. And so for these, there are a couple of specifications. So yeah, if you're submitting to Docker Hub, you have to ensure that the tool works before you upload it to Docker Hub. So that's kind of one of our stipulations. And there has to be some sort of either verification or validation that's performed for that. For GitHub, we define what it is. We have here using GitHub for version control. We have private and public repositories, and we have a little bit of a section on there, kind of going through what's private versus public, and then some links to external resources, kind of talking through you know what some of the features of GitHub are that we use typically here at the lab, and then what we want people to be able to use. All right. So now, yeah, getting a little bit more into like verification and validation. I would say that this was the probably the most difficult section to map out and to try to come up with definitions for, because when we first started this process, we kind of all got together as bioinformaticians, and then we started just kind of like listing out. We were like, okay, well, let's have two sections, one for major changes, and what would be, what would need verification, and then what would need, a validation, like a full-on validation. And so we started, listing out all the potential possibilities of when a verification was needed versus a validation. And so, we're thinking, okay, well, what if a database changes, or like, what if, the version of, a tool and a pipeline is, moved forward, or, what if you have a backwards compatibility change, and all these different things. So like we started trying to come up with all these different things, and list them out in kind of like a systematic way, and we were kind of grouping them together, and making all these different kind of assumptions and things. But it quickly ballooned, and I think in looking at it, it was kind of an untractable problem, just because you know how can you predict every single instance in which like a verification or validation is going to be needed? And so, and we quickly realized, in the first kind of drafts, like, wait, we don't want to be defining this for every single thing. We wanted it to be a little bit more general. And so we kind of decided that ultimately it was going to be too dependent on like the pipeline, or the tool itself, to come up with a rule around every possibility of updating. And so that's going to be kind of left up more to the tools themselves. And so that is kind of like where we landed in terms of the guidance that we have in this section. The general expectations, though, is that the tool must be fully developed. And so, if there's any sort of changes that are made along the way, if you're going back to dev and making any, doing any tinkering or anything like that, then you would have to kind of assess the tool in terms of like a validation. So you can't make any changes to the pipeline midway through a validation, or else you would have to go back to the beginning and start the validation over again. So, we want the tools to be in their final form. This is the final boss of this tool, before we're moving on and starting, any sort of validation. Verification is a little bit of a different thing. And so, not everything needs a full-on validation, because validations, number one, are time intensive. It can take months to get a validation through, and especially, in terms of the levels of like approval and things like that that need to go kind of up and down the chain of command. And so a verification is something where, there might be like a minor change, like a pull request message. And so in those kind of cases, what we have is a minor version verification. And so we've created kind of some documentation around verifications uh, that is less intensive than the validation documentation that we have in the laboratory. But so there's certain cases where like a if it's determined that it's like a minor change that needs a verification in order to keep using the tool, we've okayed it with our QC department in order to create these kind of documents that then map out like what it is that changed within the case, and then we have a page kind of giving the results of the test and proving that the requirement that was initially uh, asked about is actually met. And so any additional attachments like code review and logs of GitHub pull requests typically will suffice to for that proof. And so we'll just kind of like attach a screenshot of the GitHub pull request to the verification requirements proof form. Um, but so this was just a way to kind of ease the process a little bit for making sure that we didn't have to do a full on validation for every single thing, but we still have QC documentation related to verification. So for validation requirements, this is, obviously a lot more kind of intensive than the verification requirements. And so validations, these are going to be for new tools or major changes. So anything that was deemed to be, kind of like a major change, either backwards compatibility is gone or there's some sort of new functionality, there's new reporting that's coming with this. We need to be able to validate those. And so then the validation process includes a proposal, and so I've got a picture of what the kind of proposal form looks like for. So this is typically what's used in the laboratory, and we're kind of modifying this now for bioinformatics because typically there's not a whole lot of safety requirements or waste management that necessarily comes with bioinformatics. Hopefully not, at least. I hope that you know, all you bioinformaticians are safe on your computers and not dealing with, terrible safety uh, infractions. But one of the big things is having this plan for the validation. So, detailing what samples are used, the number of replicates, what analysts are going to be running these on what machines if you have multiple different configurations and things like that. What are the methods? And then also kind of looking into data retention and those policies as well. And also, of course, the resources required. So, what is the computing time expected to be? What are those costs kind of expected to be? Those kind of things. So we've had to kind of, finagle some of the wet lab documentation into a bioinformatics lens and be able to make it so that it works for us. It doesn't always, fit one to one, but we're making it work. And so then with a validation, we also have a write-up. So, we list the background of the problem, the dependencies or the requirements for the particular tool that has been developed. We, you know, have stipulated that we would like to have a workflow description. That's typically really nice to have. You have some sort of either like a subway map or a diagram or flow chart showing kind of, the different dependencies and requirements, how they flow into one another, how they interact with one another, and then what the kind of like outputs and things like that are. Pretty standard stuff. Uh, Installation procedures. it's always nice to know how you actually get it up and running. And then the completed validation plan. And so that's going to be, outlining the steps of this validation. So like, what are we doing? Notes on the problem solving and troubleshooting. So if there was any kind of like, problem during the process, were there things that we know can typically account for some of those kind of like troubleshooting errors that might pop up. Also, defining the testing data sets. So edge cases, expected outcomes, what's the scope, those kind of things. Then afterwards, there's a detailed discussion of the results. So like if there was anything that, you know, um, with our testing, if there was anything that came up that didn't necessarily, comport with what we thought would be there, why did that happen? Is that, grounds for any sort of kind of like further investigation? Or was there anything that, looked weird and why? And then we also attach an SOP or job aid for that particular tool itself. Um, and also training and competency documentation. And so that's another thing that DCLS is also kind of trying to work on right now too, is creating competency documentation around the running of the pipelines that we have within our house. So like we are trying to make sure that everybody is competent on the pipelines that they've been trained to run, and we have documentation surrounding that, showing that these people are doing this on at first a 6 month basis and then yearly afterwards, to show that yes, they still can run the pipeline, they can interpret the results competently, and they have the knowledge and know-how to be able to continue to run those pipelines or tools. Um, And then for evidence of code review, that is also something that is in our validation now, and this really came about because we wanted to make sure that anything that is being developed at DCLS has some sort of like, secondary code review. We're not just having one person kind of like write it and then send it on its way. And all code reviews can be shown through a GitHub pull request. So like we have a form that people will fill out for doing one of these kind of like, code review requests. And then they'll do a pull request on GitHub, and that will be sufficient. Like a screenshot of that is sufficient to kind of meet that requirement. All right. Um, So now getting into a lot of, so this section is probably the longest section of the document. So this is our development standard practices and expectations. So, how do we want people to think about, creating new tools at DCLS, and then how are we actually putting those into practice, and what are the expectations surrounding them? And so we have, these kind of initial, development initiation phase. So we're meeting with people who have requested that this be, created. So we're defining, what it is that this particular tool is going to address, starting to figure out the design, figuring out what is the end goal and what are these, customer asks. And so, who's asking for this pipeline and why. And then we also, in the development initiation phase, kind of talk about, what are the timelines that we're expecting to have this finished. Then the planning phase, the what, why, and how. And so this section I think is really nice because it's kind of like a sanity check for us to kind of be like, okay, well, does a tool of this nature already exist? Do we need to be creating an entirely new tool? Are we rebuilding the wheel if we're creating, something that already exists and has enough functionality to kind of get to what it is that we need in the first place? So if we can find something that already exists that works for our purposes, then we've just skipped the whole rest of this, and it's great. Or, can an existing tool be modified? Maybe something exists out there that is close to what we need, but isn't quite where there. So, can we take that and then do a little bit of zhuzhing and make it fit our needs? Fork it to our repositories. And then this section is also going to be, looking into the literature and code repositories and doing kind of the research, the background of, like what has already been done in this space. Is there anything that has been done in this space? Is there any kind of like guidance or caveats or pitfalls or things that we need to be aware of as we're going into this? And then, of course, there's the validation proposal. So I kind of, already outlined what the validation proposal entails here at DCLS. But then this is also kind of the section where mm, there's a lot of questions that are kind of posed in this section. So, we have the validation proposal template. And then, do we have any sort of like mock-ups of what we're thinking with this? This would be a section where we would kind of, start building out like a design document, which is something that I really enjoy and like in the kind of life cycle of um, code development. How will the code be tested? What machines, again, are going to be used? Which scientists are going to be completing it? How is it going to fulfill this, criteria that we have for acceptance? And then where will the data be stored? How long? What's the timeline for this? How long are we looking at? And then also, any sort of budgeting or um, requirements. Then we go into our development process and procedures. Um, and so this is, again, going to be kind of building off of like the validation proposal. So once you have the proposal kind of laid out and in the document, then you kind of go through the development process. And items from the proposal should then be turned into these kind of working documents and updated accordingly. So if there's anything that changes during the process, then we update it. One of the big things with the development process and what we tried to kind of hammer home here is that all materials should be stored in a commonly accessible platform. That's something that, sometimes I think if people are storing things on their own, personal desktops or spaces, then sometimes it can be a little bit difficult if somebody's out or somebody else needs to kind of like take over a project, to find all the information and things that are there. So we have some commonly accessible storage options on like AWS and things like that. But then there's also GitHub too, which is everyone within our organization has access. And this, for the development process, if there are multiple developers who are involved in it, we tried to stipulate that it should be, part of the process that there are regular meetings to collect feedback and discuss kind of the progress. And then, of course, we have, our GitHub repos, and we have a few kind of things kind of laid out here in order to talk about, how do we create repos and branches and licenses? When we make a change, what are the different kind of procedures that are involved in that? And then, what is required to be under any sort of GitHub that is created at DCLS. And so one of the big things that I think a lot of people really appreciate is having a README, that will describe the tools, authors, instructions for installation and use, and then it should be maintained throughout the project. We go into branch management. So, and I'll get into this a little bit later because in some of the kind of like appendix sections, there's, I think, a really nice diagram that kind of shows, how we're trying our best to kind of like adhere to branch management and, changes and patches and things like that. And of course, with this one, all code is going to be versioned according to that, semantic versioning that was presented a couple of slides ago. So we have our pre-coding phase, our coding phase, and then the reporting phase. So mockups and plans of execution, we propose the test data, then iterate through. With the coding phase, drafting, testing, troubleshooting, and then of course iteration. So, doing it over again. So once we're finished, the validation plan is updated, and a final validation package is put together. And then once everything is kind of signed off with the we're good with the validation, then we do the code review, which is that pull request. And it must be reviewed by a separate bioinformatician from the one who pull requested. So that is a stipulation in the document. You can't code review your own code. All right. Um, So these are the questions that are asked in the code review, and so this is what we're putting kind of in the GitHub pull requests. So does the code appropriately and effectively address its purpose? Does the code follow DCLS's security guidance and practices? Does it follow efficient logic? Is the process or code readable and commented appropriately? Is the code portable? Are there hard-coded paths, or are there containers and workflow managers used? Are reputable dependencies used? And does the updated verification or validation plan for the code effectively assess the following when applicable? The resources, processes, or code is used in a manner that appears to generate correct data, and the data generated from a custom calculation or function is usable, factual, and dependable. So this is kind of, we have this little template, and then people, who are doing the code review will go through and address these different questions. So this is kind of the final section of the document, and this is one that I think is probably going to continue to go through some iterations. But so for the components of pipelines in there, the recommended updating schedule. So we're we're trying to come up with like a minimum schedule for updating and looking at, when things need to be updated. And so we have, some of these different things: programming languages, packages and libraries or modules, data source, and workflow managers, command line tools. Um, a lot of these are still kind of like in the phase where we're trying to kind of document everything, make sure that we have people who are dedicated to actually like reviewing this on a specific schedule. And so that is some we have some documentation that is being drafted up and worked on for this right now. And so that is kind of where that process is at. We also have in the tool maintenance uh, section this kind of like scheduled update review procedure. And so then if an update will introduce issues with an existing code, a verification or validation must be performed on update. And then we have this kind of color coding of urgency for categories for prioritization. So trying to make sure that we're getting to the things that need to be updated most immediately first, and then kind of going through and making sure that everything else does get updated if it needs to be. But if it can wait, then or if it doesn't need to be updated immediately, then it's lower down on the priority list. And so this is in the appendices. And so this is, I think, a really nice diagram for branch management. And so this is kind of what we're trying to adhere to. And so that's kind of, talking through what the semantic versioning looks like, what these kind of branches would look like for different features and things like that. You've got a development branch that you're making changes to, and then how does that relate to the main branch? And how are kind of like, bug fixes or patches being applied off of that as well. And one of the other things that we have in our appendices was information which should be excluded from GitHub, oops, for security best practices. And so, this was kind of, this is in direct compliance with kind of our laboratory QA department and kind of some of the security that IT is overseeing as well. So no IP addresses should be uploaded to GitHub. Obviously, no PHI or PII. Personal user information, passwords and things like that should not be uploaded. Any proprietary information that DCLS owns, um, information related to ongoing litigation, and then any information that has been expressly deemed inappropriate for sharing by leadership or IT. And Yeah. All right. So this is kind of my last slide, I think, of the presentation. And so, I wanted to end this with the fact that like this is, I think, a really great start for us, and this is something that we're really proud of, and I think that we, did a lot of work to try to come up with something that would be engaging and useful and hopefully, inform some of our kind of practices here at the lab. But as with everything, there are ongoing challenges and there are things that we're going to have to address kind of going forward, I think, too. And so the enforcement of this, I think, a lot of this is kind of you know, self-enforced right now. We're trying to come up with ways to keep things on, schedules, keep things, so that they're being looked at on, particular timelines and things like that, and making sure that anybody who is working on it, it knows what the expectations and the practices are, and then, adheres to them. But that's always going to be a challenge, I think. Familiarizing ourselves with the document, and so making sure that new people who are brought on are familiar with what this document entails, and everything that we expect as an employee at DCLS. And then I think this is a good first pass, and I think that we did a pretty good job. But with everything, there's always going to be updates. There's always going to be things that need to be changed, and so I think that this document is going to be, you know, hopefully kind of like a living document as either practices change or things kind of update with the lab infrastructure. And then for the definition of specifics, like I said, we tried not to get super duper specific with this particular document, but I think that we're trying to say that those kind of details are going to be more useful to have in like the specific SOPs and the specific job aids that are developed for the particular tools which they're corresponding to. And so those are kind of some of the ongoing challenges that I see, and kind of some of the things that um, we're working through. And I think that's it. So we've got a little bit of time now, I think, for questions. Um, and one thing I did want to say is I know that there are a lot of other labs who are working on kind of like similar documents, and there are people who have developed similar things, and I'm really curious to hear how other people are kind of approaching this. One of the reasons that I think I'm giving this talk is because at APHL annual this year, a lot of people were kind of in the same boat and thinking about like, how do we kind of move forward in this kind of bioinformatics regulation and kind of that space. And so I'm hoping that there will be some questions and maybe some discussion.
Go ahead, Kelly.
Yeah, no, that was awesome, Logan. And I think everything you've done is really well thought through and outlined really, really well. I guess I had a question. Um, in terms of like CLIA, do you consider your bioinformaticians CLIA testing personnel, or is it more on the wet lab staff who actually then run the pipelines? Are they considered the testing personnel that need to be then considered competent for running the workflows?
That is a good question. For the CLIA pipeline, like we don't typically right now we're not necessarily explicitly trying to CLIA validate all of our pipelines, but anything that would be CLIA validated, I want to say I need to make sure that I'm giving the correct information, but I think that the people who are running the pipeline are considered CLIA personnel in that particular instance, and that's why we're doing like the competencies and things like that, and making sure that they have documentation showing that they are capable to run the pipelines and that they have, actual experience with interpreting the results that come out of it. But that is a tough question.
Yeah, it is, right? Something that we keep getting asked every single time we get inspected, right, is, well, shouldn't your bioinformaticians be testing personnel? And we say no, because they're not actually doing anything or interacting really with the results, right? They're ensuring that the work that, the computational side works, but they don't actually do anything with the data as we're running everything in an automated fashion, right? So as sequencing runs get finished, our workflows kick off, automatically. We don't have to actually have somebody go, okay, run, stop, okay, run, stop, right? It's just they're just running, right? And so um, I'm just curious, right? Because keeping bioinformaticians competent has kind of been a struggle for a lot of labs, right? Like, how do you actually determine if a bioinformatician is, in the CLIA definition, competent to be doing what they're doing, right? How do you test that competency? And it's been something I know, we've been struggling with.
Yeah, we do have some kind of forms and things like that that we're trying to create through our QC department. And then we have like a schedule that we're keeping people on now. So like any tool that is created here, we have a competency form to go with it, and then we have kind of like a series of steps that they follow,
Right.
on
But do you have
a
proficiency testing
timeline.
for,
So
for them in place that happens twice a year?
we have it's the competency testing. So yeah, I don't think that it necessarily would be the same as the proficiency testing.
Right. Because like any CLIA validated test, right, you've got to run that, at least twice a year, some kind of proficiency testing, either an internal one or an external one to show that your testing personnel are proficient and competent in performing that test, right? So we've always struggled like, well, how do we give one of those to bioinformaticians, right? Who really, yeah, anyway, it's just a question that we always kind of have to dance around with CLIA is like, well, we don't list them as testing personnel, so we don't have to keep them competent or proficient. So yeah, that's always been kind of our answer. So I was just curious how you guys were handling it.
I think for a lot of our pipelines, they're not CLIA validated, and so we're looking at, like what would it take to be CLIA validated, or like what kind of things do we need to put in place in order to ensure that we could meet those requirements if need be. But at the moment we're kind of shying away from that, like stepping up to the line, but not necessarily crossing it.
Cool. Yeah, no, that totally makes sense. Thank you.
Oh, Christian.
Yeah, to answer your question, and Logan, I don't know if this is the same in for your side of bioinformatics, but at DCLS for newborn screening, we participate in the annual or biannual PT testing. And as part of that analysis, it goes through our bioinformatics pipeline as part of the assay and the testing. So, that's how we take part in our PT testing as far as meeting that clear requirement for newborn screening. And I would assume so. I think talking to Molly, she said for like the infectious disease and foodborne PT testing, it goes through the bioinformatics pipeline and interpretation. So as part of that kind of umbrella, yes.
But you have your wet lab staff running the bioinformatic pipelines, correct?
No. Um,
So you have a bioinformatician that runs the
mhm.
pipeline. So how
Yeah,
do
we
you
have a
keep that person competent? Because that's a question we get asked, right? So we told them, well, yeah, we'll just run the Enterix pipeline, right? We'll get those
mhm.
PTs from PulseNet, we'll run them through, and that should make our bioinformaticians competent. And we were told, no, that doesn't work.
Well, we do our annual competency paperwork
Right,
for the
right,
bioinformatics pipeline on top of the PT.
right. But I guess what I'm asking is, so we got inspected by CLIA,
Mhm.
and we said, okay, we had two bioinformaticians. They wrote the pipeline to do the enteric pathogen data analysis and call serotype. We said, okay, cool. They're like, well, how do you keep them proficient? We said, well, we run, right, PulseNet's enteric pathogen PT. We've got a PT program that we do with Minnesota, and they do that. And they're like, well, that's not enough to keep your bioinformaticians proficient.
Yeah, I'm surprised by that response too. Because we're meeting the same um requirements of the wet lab in terms of our competencies and proficiencies, and that we participate in the biannual proficiency, and then we do our annual competencies on each bioinformatic pipeline that we perform.
Yeah, so we must have just gotten this.
Yeah,
Yeah. Hey, Kelly.
I'm surprised by that.
Yeah, well, we were too.
Yeah, I was curious, Kelly, did they give you any explanation for why they didn't think it was sufficient?
Um, they didn't like it because the person that was running the pipeline was also the person that had developed the pipeline. And they wanted to know, somebody's developing a pipeline used for a CLIA test. How are you keeping them competent on the development side?
Wait, competent on the development side. That doesn't make any
How
sense.
do you, sorry, I'm using competency and proficiency in probably interchangeable ways here. How do you make sure that somebody is proficient at developing a CLIA validated workflow?
But that
Was
seems like it defeats
the
the
question I
purpose
got
because we're
asked.
talking about production.
Right. And this is where I said we probably just had a really crappy inspector that go around. So what we did is said, okay, they're not testing personnel. Our bioinformaticians are not testing personnel, and we made it so they don't interact with any of the data that comes out of a CLIA validated workflow to get around that question.
But
Because they actually cited us for that, is that our bioinformaticians weren't technically part of the proficiency testing algorithm. So therefore they weren't being kept proficient. Therefore that's a validate or a violation of the CLIA. So actually got written up for that. And our response was, cool, well, we'll make this pipeline automated so our bioinformaticians never have to touch it. That way they don't have to be proficient. We also got cited because they didn't take one of my bioinformaticians um, uh, transcripts as being enough information to make them CLIA testing personnel. Even though this person had a PhD in biology, it was in cancer genomics, they didn't believe it. They couldn't interpret the transcript to say, yep, that person's got enough biology credits. So it was a really bad inspection that year. But so this is why I'm always curious about others' inspections to see how they've dealt with those issues that may have cropped up.
Kelly, are you guys using these results? Are you sending them back, and using them for clinical purposes?
For Salmonella and E. coli, we had providers that were requesting those serotype results be sent back to them. And so we CLIA validated it so we could get rid of running, another test for Salmonella and E. coli serotypes. And then on top of that, we validated an oxygene family whole genome sequencing result that goes out for carbapenem-resistant Acinetobacter baumannii. We've also done Vibrio typing and Shigella typing as well under CLIA. So yeah, it's been fun. But we got inspected like, a couple months ago and we were just fine. So it's hit or miss sometimes with bioinformaticians and CLIA inspectors, I'll say.
Yeah, that sounds like it was just a tough inspection.
Yeah, and like the guy just didn't want to listen. Like this is the same person that said, well, if you're using base space, how do you know that Illumina is not manipulating your data and changing it before you download it? Yeah.
I see Molly has her hand up.
Yeah, hi Logan. So what you presented is like pretty similar to what we're doing in Colorado, but I did have like two questions that are somewhat unrelated. Like the questions themselves are somewhat unrelated, but they're two things that we've kind of struggled with in Colorado. So my first question is like, how do you choose the data set that you're going to test the data on? Is there like a standardized data set, or do you specifically choose a data set that has exceptions that are either going to break your code or it's not going to break your code? So that's kind of the first question I have. And then the second question I have is with the development standards and practices expectations, like you have that planning phase. Like how much of that is like a group effort versus an individual effort?
Yeah, so I can go into I'll go into that question first, and then I'll go into the data sets, I think. So with the planning phase, I would say that typically it is more of a group effort at first. So like if there is a need for a pipeline, we'll kind of like talk through it. Just to see like, okay, what is it that's actually needed here? Most of the time, once we're talking through something like that, then somebody will have found either something that already exists, and so we can say, oh well, let's use this particular thing instead. If not, then it's kind of like assigned to a person, and then that person will then kind of like take over most of the development and the proposal and kind of the downstream things. But we do try to be somewhat, I would say, collaborative, and especially like depending on who's asking for this particular pipeline as well. So it could be that it's an internal request, and so maybe this is something that the wet lab is asking for, or it could be like an external request too. And so like if this is something that EPIs are asking for, then we just want to make sure that everybody is coming to the table and has kind of a say, and we all know what is the end product that we're actually trying to create before we kind of get started with that, because otherwise we've gotten into situations before where you start developing something that maybe there isn't quite a need for, or you come back to the table and people go like, oh well, that wasn't what we wanted. For the first question that you had with the data set choices, yeah, data sets can be really difficult, and so we will try to come up with you know, I think a lot of the time what we're looking at is a specific kind of like feature. So like we're if it's like a characterization pipeline, like we'll try to get as much of the breadth of scope of the project that we're trying to look at with the data set, but we will choose those ourselves. And I want to say I'm blanking on if there's an actual like number of samples that we had to test, and I think that there is some guidance around that, and I need to familiarize myself with that and find that for you. But we do have some kind of like already pre-established data sets, and so like for kind of when we're doing like a verification or something like that, there will be like the data set that we've used previously for the validation, and then we'll typically use that again to rerun the data.
Thanks, Logan.
All right, well, we're a little past noon here, so there's no more questions for Logan. Thank you so much for doing the talk, and thank you all for joining us. Uh, have a good afternoon.
Thanks, everyone.